So GMOs are regulated in New Zealand under the Hazardous Substances and New Organism Act, otherwise known as the HASNO Act. This was brought into force in 1996, although the the New Organisms provisions did not go into effect until 1998, and they did not fully take effect until 2001 with a 3-year transitory period. This act gives the EPA the authority to determine whether any organism, and I'm not just talking about GMOs here, I'm talking about any organism at all, is new to New Zealand or not. So the fundamental underlying principle here was about the preservation of New Zealand's native biodiversity because of some of the environmental problems that have been caused by bringing in kangaroos and possums and things like that, and rats and stoats into New Zealand in the past. The genetic modification part of it, I think, came a bit later, but it was right around the same time as the Act was being brought into force. So, new organisms and their status in New Zealand. Any organism not present in New Zealand on or immediately before 29th of July 1998, or any genetically modified organism, regardless of when it was created, are illegal in New Zealand without EPA approval to be held in containment or for release. So the question of what a GMO in New Zealand has to do, it's rather similar to what we would have under the Cartagena Protocol, to which New Zealand is also a signatory. All GMOs are defined as anything that results from the use of in vitro manipulated nucleic acid. Now, most of the time this means DNA, but obviously some organisms have RNA as their genetic material, so that counts as well under the Act. So these are considered to be new organisms except where they're exempted in regulation. Now, some of these regulations have nothing to do with in vitro techniques, but I believe they were put in place at the time to give everyone surety and understanding of what actually is exempted. So I'll just go through these rather quickly. Selective breeding, natural regeneration, say of plant shoots, things like that, and hand-pollinated are exempted. This is obviously not in vitro. Any regeneration from organs, tissues, or cells, somoclonal variants, and cell fusion. And this is one area where New Zealand slightly differs from the Cartagena Protocol on what's allowed and what's not. The Cartagena Protocol considers a GMO to be something that results from cell fusion outside the taxonomic family, whereas in New Zealand, any cell fusion is considered not to be GMO. And we deal with this with the LMO Prohibition Order 2005 in New Zealand, which is another law that we have to ensure our compliance with Cartagena. Other than that, it's essentially complete overlap with Cartagena. This third one is probably the most important one that I'd like you to take note of, because this has actually been the source of a court case based on a determination that EPA did. So chemical and radiation mutagenesis causing chromosomal rearrangements was what was exempted in 1996. As I say, I'll come back to this. That has been the subject of a high court case. Artificial insemination, superovulation, embryo transfer or splitting are also exempted. Physiological nucleic acid movement, i.e. conjugation of plasmids and so forth. If you're doing that in a petri dish, that's fine, unless, of course, you have been using in vitro manipulated nucleic acid prior to that, in which case it's not exempted. Any spontaneous deletions or rearrangements of DNA, of the genome, are also exempted and can be used. So to come back to the issue regarding the chemical mutagenesis aspect of things, the not GM regulations were brought into focus or into consideration with regard to site-directed nuclease technologies starting in 2012. And this had to do with what at the time were the cutting-edge techniques, zinc finger nucleases and transcriptional activator-like effector nucleases, otherwise known as TALENs. CRISPR-Cas was just barely on the horizon at this stage, so it wasn't really part of the consideration here. So this was applied for by one of the Crown Research Institutes in New Zealand as to whether or not certain applications of this technique would be considered not to be GMOs. In other words, to use the proteins, so with zinc finger nucleases, you can create a custom recognition, DNA sequence recognition site, and you can then attach that to through genetic modification means to a site-specific nuclease or a general nuclease such as FOC1, and that's also being done with talons with the tail repeats. And the question that the EPA was asked to consider was whether or not if these were used as proteins with cells to actually create mutations. So the cleavage can sometimes be used to simply just to fix itself. Sometimes that goes back to the wild type sequence. Sometimes that makes a change. So if you know the sequence, you know the effect of your mutation, it's a very useful thing to apply. And it was hoped by one of the research institutes, specifically Scion. They have a lot of issues with long breeding times in forestry, so they saw this as potentially a way to get to specific effects in trees much more quickly. So they asked us about this. And that it was decided at the time that these techniques, when used as proteins and don't involve any nucleic acids into the organism that you're modifying, the EPA actually found that that did not constitute genetic modification. This was almost immediately challenged in court, and there was several months of hearings in 2013 in the New Zealand High Court. The judge considered the arguments on both sides and decided that the EPA was, the decision was quashed. Not because the judge actually viewed these techniques as creating genetic modifications. It's simply that our regulations require that any exemptions be very explicitly put on a list. And since these were not on the list, they had to be considered to be GMOs. This had some ramifications further downstream, because if we go back to the to the bit that I was pointing out to you in terms of the exemptions, what you'll see is it says chemical and radiation mutagenesis causing chromosomal rearrangements. Now, point mutations, which is what the subject of the SDN1 determination was, are not chromosomal rearrangements. And therefore, this ruling called into question any technique that created point mutations, because that's not specifically on the list. So this precipitated a 2-year process of reviewing these regulations. And we undertook an extensive review of all the technologies, the EPA along with the Ministry for the Environment. We looked at these technologies in the context of the regulations and conducted public consultation. Now, there was some concern around potential trade issues, because New Zealand at the time would have actually been first to say that these techniques did not create GMOs. And so the concerns around trade meant that a conservative approach was actually taken to the consultation. So this was put out to suggest that basically what we needed to do was to fix the language to ensure that point mutations in the traditional sense were exempt. Otherwise, essentially all the crop plants that are grown in New Zealand were potentially being illegally grown in New Zealand. And also to reestablish the status quo of 1998. And the advice to the minister at that time was that this was probably a short-term fix because the technologies are advancing quite rapidly. So in 2006, after this consultation, we received, I believe, 39 submissions. Most of these generally agreed with the proposed changes, and most of the submissions actually suggested that the changes did not go far enough, and those submissions came from industry and research groups in New Zealand. Regardless of that, the regulatory changes were made to say that any technique used for mutagenesis that was not in use or invented before 29 July 1998, which is the date of the new organisms regulations went into effect to begin with, these are now considered to create organisms that are GMOs. So, just to change gears a little bit, I just wanted to tell you a little bit about what is actually going on in terms of activity in New Zealand, and we do have a couple of approvals for field trials, although the definition of field trial in New Zealand might be a little bit different from what you would be used to thinking about in general, in that the field trial is considered to be outdoor containment. And what that actually means is that no heritable material may escape the field trial. So in the case of large animals, you can breed those animals because they are actually confined to a field, and they're not that hard to keep track of. Whereas with plants, pollen flies, this pollen can be carried around by bees, taken to plants that are not part of the field trial, or in the case of pine, this is all borne on the wind. And so there's some difficulties with doing some of the field trial work with plants, because you can't actually grow the plants to the stage where the most economically relevant traits can actually be tested. And particularly with the case of pines, the trees do not really develop their wood quality traits that you can actually observe until they're actually able to set seed. And so it makes it very hard to actually test some of these things in those instances. So just to go through this briefly, there is one field trial approval with Ag Research in Hamilton for cattle, goats, and sheep. Currently, there's only cattle and goats that are being tested. But some of the traits that they're looking at there, for example, high casein milk in cows, hypoallergenic milk that could potentially be used in infant formulas and so forth, so beta-lactoglobulin knockdowns, and biofarming, so monoclonal antibody production in the milk of cattle and goats. With the field trial of radiata pine, this was approved in 2010, superseding some previous approvals that were essentially about environmental effects, and now they are testing or attempting to test commercially relevant traits such as wood density, growth rate, and other wood quality traits such as strength, stiffness, dimensional stability, in other words, resistance to warping. Regarding releases, we have two in the 18 years of the Has-No Act in New Zealand. And one of those is quite recent, and that is for a genetically modified oncolytic vaccinia virus liver cancer therapy. So it's called Pexivec. It's part of a phase 3 global clinical trial. It is a genetically modified vaccinia virus, and vaccinia was used in the eradication of smallpox. So it is does not cause disease in humans, but it is cross-protective there. And so this has been used to make a virus that will actually attack tumor cells. And the approval here is for a conditional release in that let me back up just a moment here. So what we do in terms of approvals at the EPA is not to actually approve or disapprove a medicine as such. What we do is an environmental assessment, environmental risk assessment on this. So we often take information about how this might affect the patient, which we're actually not allowed to consider, but translate that to how that might affect people around the patient as part of the environmental risk assessment. And so with vaccinia virus, there is the possibility, when that's injected, it sometimes causes a bit of pustule formation on people's skin, which then can be transmitted to other people. So the controls that we placed on this, there are no cases of environmental harm, but you don't necessarily want to be getting this if you don't need it. And so we do require that people are bandaged, and those bandages then are put in biosafety containers and returned to the hospital. This must be done by a qualified medical practitioner at a hospital and so forth. So it's relatively mild controls, but there are controls nonetheless. The other approval is for a genetically modified equine flu vaccine. This is called Protect Flu, which is for horses. This was approved in 2008, but until quite recently had never been used, because part of the conditions of that approval was that it could only be used in New Zealand if there's actually an emergency of equine flu, or if a country that specifically requires this particular vaccine would require it for horses for export. That's recently been changed, that the importing country might require vaccination, but is not specific as to what vaccine might be used. So that is now being used as well. So essentially, to summarize what I've been telling you here, New Zealand really is taking a wait-and-see sort of approach to the regulation of these new breeding technologies and gene editing, but it does have working pathways for the use of GMOs as medicines and veterinary medicines. And this slide here is really just to show you the reason for some of that caution is that the bulk of New Zealand's economy is based on primary sector. So dairy is the biggest industry in New Zealand. Forestry is about the third biggest, and you can see that horticultural crops and so forth are also very important to New Zealand. So there is a bit of caution there, and but I think the country will be able to adapt if things actually change in the future. Right, so one thing I do want to tell you briefly about is the EPA also has the power to do determinations as to whether or not something is in fact genetically modified based on some of the definitions of what a GMO actually is. So replication defective viral vectors, which essentially don't have the replicative functions of a vector, of a virus, have been considered not to be organisms. So this includes retroviruses, including HIV, adenovirus, and adeno-associated virus. These are very commonly used as delivery systems that can be packaged into a viral capsid or envelope and then delivered to the cells, and are being used a lot in gene therapy research. So the further step from this is that since these vectors are not considered to be organisms, animals that are treated with them, and there are a number of animal models for gene therapy research, animals that are treated with these vectors, provided this does not get into the animal's germline and make it possible to pass on to progeny, then they're not considered GMOs either. And this has enabled gene therapy research on large animals that don't require containment, which can be very impractical. Okay, just to finish, a couple of new developments that are going on. Last year, New Zealand introduced, the government introduced what it called the Predator-Free 2050 initiative, in which it's putting forward $28 million to attempt to rid New Zealand of rats, stoats, and possums by the year 2050. So these organisms are perhaps the worst destroyers of biodiversity in New Zealand, both for plants and for other animals, and they are targeted. One of the goals here is that there should be a new technology that is specific for killing one of these predators. A promising one currently is not a GMO, but actually a rat-specific poison, which a lot of research is going on with now to not change its specificity, but to make it a little bit slower in terms of its effectiveness to ensure that the animal eats enough of it before it actually is killed, rather than get sick and never goes back to the bait. So I've provided you with a couple of links to that, just so you can get some further details on that. This has been amplified just this year. Excuse me. So New Zealand's Parliamentary Commissioner for the Environment has issued a report on the conservation status of our native birds. And New Zealand is a strange place. You might know that Australia has a number of mammals that are found no place else. They're marsupials. In New Zealand, there are no native mammals, except for 3, and they're bats. And they've been thought to have flown over from Australia at some time in the past. So essentially, all the large animals, or larger animals, were birds, and many of them, because of lack of predatory pressures, lost the ability to fly and tended to nest on the ground. And when mammalian predators were brought in, this has been an absolute disaster for New Zealand's bird species. So 80% of New Zealand's native birds are currently under threat. This mostly comes from invasive mammals, and one-third of these bird species are under imminent danger of extinction. So the Parliamentary Commissioner for the Environment has put out an emergency call, really, to say it is time to actually start to consider some genetic technologies for the conservation of these bird species. And she's instructed the ministers of several government departments to begin to educate the public on what these sorts of technologies might be useful for. So among these, obviously, are gene drive, and there have been a number of discussions in New Zealand already, not by the government, but by others there, to say that gene drives to essentially make all the progeny male and so forth as a humane way to eliminate predators without causing them any pain or anything like that. So there hasn't really been anything coming from the government so far. This is primarily because we're having an election in about 2 weeks, and so that's something that nobody's wanting to touch at the moment. But I think that things are going to get very interesting before the end of the year. And I would like to thank you for listening to this. And if you have any questions, I'll be happy to talk to you at lunch or sometime during the conference. Thank you.